Hi, year 11s and 12s. Today we're looking at Unit 1.4, where we're looking at mutations. This is one PowerPoint. There's going to be two videos this week. The first one's looking at the different types of mutations that occur in the body and the different types of mutations that genes will experience. Whereas the second video is going to be looking more at the mutation rate. How mutation rate can be changed throughout different circumstances. So let's begin. As per usual, we've got our need to know. So again, I'm not going to go through these. If you'd like to write them down, this is where you pause the video, take them all down. But again, only about 5 points covered in this PowerPoint across the two videos. Okay, so first off, let's actually talk about what a mutation is. And when we're defining a mutation, we refer to it as a sudden change in the sequence of DNA. Sometimes mutations are also referred to as point mutations because that's a very specific type of mutation. A point mutation is basically where one nucleotide base in the entire DNA strand has been changed. That is enough of a change in the DNA sequence to change the mRNA codon, which will then code for a different amino acid, which will change the primary structure of a protein, which could affect the entire protein. So all you need for a mutation to actually cause damage is for one nucleotide base in the entire DNA sequence to change. There are 4 different types of point mutations that we need to talk about. So there's deletion, insertion, substitution, and inversion. Now, generally, these 4 different types of mutations occur during DNA replication. We're going to look at, to describe how they work, we're going to look at this strand of DNA, ACT, GGT, AAT, GCC. Okay, so we're looking at this strand of DNA, and we're going to apply each of these types of mutation to it to show what would actually happen. So, in deletion, physically, in DNA replication, one of those nucleotides is deleted, so it has a mean copied over. So, instead of having ACT, GGT, we've got AC, nothing, GGT, AAT, GCC. So, when this was being read for codons, it's more likely going to end up being ACG, GTA, ATG, and then we wouldn't have a full set of codons for the last one. That's then changed every codon from the point of that deletion. So that could cause serious changes to the protein because potentially every amino acid after the deletion has been changed. We also have insertions. So in this case, we've inserted an A. An extra nucleotide's been added during DNA replications. Now we've got ACT, AGG, TAA, TCC, and then we have one nucleotide left to start a new codon, but nothing else. Okay, so again, this has shifted the entire, every codon, basically by one, from the point of the insertion. So again, major changes. Substitution, on the other hand, is only going to be one single mutation. So in this case, we've substituted an A for a T, and it's only going to affect the codon that the substitution occurs in. So instead of ACT, we now have got TCT. So that one amino acid could potentially change, which still will cause massive errors and massive problems to the protein because all of a sudden it can't fold the same way because it's got a different sequence of amino acids. That primary structure has changed. Inversion, again, so inversion's only going to affect the codon where the mutation occurs. So in this case, A and C have physically been swapped around, so instead of ACT, we've got CAT. So they're the 4 different types, and how they affect, basically, the protein structure. So deletion and insertion will affect every codon and every amino acid after the deletion or insertion, whereas substitution and inversion will only affect the codons where the mutation actually occurs. Like we've got types of mutations for DNA, there's also different types of gene mutations. The different types of gene mutations that we're going to look at are Missense, Samesense, Nonsense, Frameshift, Chromosome mutation, as well as Aneuploidy and Polyploidy. Now, we're going to talk about each of these on a different slide. Each of these has the potential to change the secondary, tertiary, and quaternary structures because they're changing the primary structure. Okay, let's start having a look at those. So first we're going to look at missense. So a missense mutation results in a different amino acid being coded for. So over here, while we've got CAT, what's happened is we've had a replacement, so a substitution point mutation occur, which is instead of CAT, we've now had CCT. And what that's done is it's changed the amino acid here, HIS, into the amino acid PRO. It's no longer making sense because that protein doesn't make sense anymore because a different amino acid is there. This entire protein's primary structure has changed because of that one amino acid swap, which is going to cause a change in the second, tertiary, and quaternary structures. So, missense generally is only a change in one codon, so this is generally going to be caused by substitution or inversion. We also have a samesense mutations. So, these are still mutations, okay? So, they're a gene mutation in which the codon has changed, but the amino acid hasn't. So, for over here, we've got a mutation. Instead of UUU, we've had UUC. The amino acid hasn't physically changed, which means the protein hasn't changed. This mutation is silent. So we call them same-sense mutations or silent mutations. Nonsense mutations are interesting. So again, this could be caused by anything. This could be caused by inversion or substitution if it was just a straight swap. But it could also be caused by insertion or deletion because we're not worrying about anything beyond the mutation. So a nonsense mutation is a mutation that what happens is the codon changes to be a stop codon. So it stops the entire protein being produced earlier than it should. So in this case, instead of having our 7 amino acid protein chain, or longer because we can't see the rest of it, we've ended up with a 3 chain amino acid protein. Because the stop codon has been changed from the 4th amino acid into a stop codon. Okay, that's nonsense, exactly as it sounds, because the protein doesn't make sense. It's been stopped at an incorrect point. Because it's a lot shorter, that's going to dramatically change the secondary, tertiary, and quaternary structures, and mean that it's very unlikely that that protein is going to have a biological function. Frameshift mutations, these are when the gene undergoes specifically an insertion or a deletion, which results in one or more of the codons changing. So, as we talked about on the earlier slide, an insertion or deletion is basically going to change every codon from that mutation onwards, which can essentially change every amino acids after that mutation. Okay. When we have large changes like this, it's very likely that it's going to prevent the protein from folding into a shape that's biologically active. Okay, so just trying to find out where we've actually got. Okay, so what's happened? Somewhere along the line, we've had a mutation that's an insertion or deletion beyond what we can see in this picture, which has changed. Instead of CAT being the codon for this amino acid, we're now looking at ATT, which has changed every amino acid going along. There's an entire frame shift in this amino acid chain. Chromosome mutations. This is a change in the number or structure of chromosomes in a cell. Generally, this is going to be caused by errors during cell division, and that can include DNA replication. Generally, when we have a chromosome mutation, it's going to change the entire chromosome, and that's going to affect hundreds and thousands of genes. So there's 4 different types of chromosome mutations. We're going to look at those just on this slide. We're not going to look at them in greater detail. So we've got translocation. This is when genes are exchanged between non-homologous chromosomes. Okay, so that's generally during meiosis, which we'll look at a little bit more in cell division. So genes are physically exchanged between the wrong chromosomes. Duplication is when a gene is replicated more than once, so the chromosome has more than one copy of that gene, or more the copies that it needs of that gene. Deletion is when a gene is deleted or removed from any location on a chromosome, where inversion is a section of the chromosome breaking off, and that broken segment being reversed and connected back up. So basically the gene is backwards. Lastly, we're talking about aneuploidy and polyploidy. So these are types of chromosome mutations where you have an abnormal number of chromosomes. Okay, aneuploidy is an abnormal number. So for example, there's 3 examples we're looking at. So we've got Klinefelter syndrome, in which males have an extra X chromosome. So they'll have XXY, they've got one extra chromosome. Turner syndrome is where females only have one X chromosome. So they've only got 45 chromosomes in total. So Klinefelter ends up with 47 chromosomes, Turner syndrome with 45, and then Down syndrome, which is another type of aneuploidy, one that's talked about more often, where chromosome 21 has 2 copies. So again, 47 chromosomes here. Polyploidy, on the other hand, is a chromosome mutation that results in the individual or the organism having more than 2 sets of chromosomes. Very common in plants with genetic engineering, not so much common in humans because generally you can't survive with having more than 2 sets. So we're talking about, instead of having 46, we're probably talking along the lines of 69 chromosomes or more. Okay, which just wouldn't work in the human body considering the effects of Down syndrome at just having an extra copy at number 21.